Welcome to Wednesday night at the Genome. I'm Celeste Berg, and I'm standing in for Elhanan Borenstein, who is the organizer of this year's summer public lecture series. Wednesday night at the Genome is in its 10th year, a fine tradition of the department sharing its exciting findings with the public. Tonight, many of us faculty are here in the audience. We'll be wearing name tags, which I just took off with my hair. And we're planning to stay after the talk along with our honored speaker to be able to answer any questions you might have about general genomic-y kinds of things. So please stick around for the cookies and for questions. Okay. It's my pleasure to introduce tonight's speaker, Dr. Doug Fowler. He is currently an assistant professor in the Department of Genome Sciences, but I fully expect that very shortly he will be an associate professor with tenure. Yeah.
We're not there yet.
Got to embarrass you a little bit. Wait till the end. Okay. So Doug earned his bachelor's degree in chemistry and biochemistry at Northwestern University in Chicago. He then moved to the Scripps Research Institute in San Diego for his PhD work on amyloid proteins. Now, many of you probably know that amyloid proteins are often found in aggregates in the brains of patients suffering from Alzheimer's disease. Doug's studies in San Diego focused his interests on the very important problem of understanding how the exact sequence of amino acids, the 20 subunits that make up all the proteins in our body, how those exact sequences contribute to the structure and function of a protein. Okay, who can tell me what a protein is? Come on, you gotta have some volunteers. Who can tell me what a protein is? A protein is something that you see on the side of a soup can, right? Right? It's an ingredient. Okay, we got an answer here.
It's a mix of amino acids that the cell has created in order to perform cell function.
It's a mix of amino acids that the cell has created in order to perform a function. That is a really good explanation. Proteins are machines, they're factory workers. They are the things that make our cells work, keep our entire bodies running. They are the substance of us. They are everything. And Doug is setting out to solve a really, really important problem. And that is that changing a single amino acid in a protein can have huge consequences. It can give us different skin colors and different hair colors. It can make some of us great athletes or musicians or doctors or people who can taste things in wine. Not me.
Truly impressive feat.
Truly impressive feat, I agree. Okay. But single amino acid changes can also cause cancer or heart disease or Alzheimer's. Right? Okay, so as a postdoctoral fellow, Doug moved up here to Seattle to work with Stan Fields, and he set out to develop a new technology that would let him evaluate the importance of each amino acid at each position in a protein. A very difficult task. But solving that task would help us evaluate how variations in our proteins contribute to what makes us us and what makes us go wrong or bad or good or whatever, okay? And Doug was so successful at developing this new technology that we quickly realized we had a gem in our midst. Yeah. And Doug decided he liked it here too. And so after competing with hundreds of other postdoctoral fellows from all over the country, we offered Doug a job. But we're the lucky ones. We are really lucky. Doug's expertise, his creativity, and rigor has attracted many collaborators who come knocking on his door to profit from this great method that he's developed. And the funding agencies have recognized the power of his methods, and he's garnered many awards and fellowships and grants. If you look at his CV, I think my favorite one is called One Brave Idea. Sounds kind of sci-fi to me. Okay. Now, Doug's done extremely well. He's attracted a lot of talented students. He's garnered accolades from his teaching. He's really done quite well. But that doesn't mean he hasn't suffered from beginning faculty syndrome. We all can tell when Doug gets really stressed out because he pulls his hair out. He has this sort of hand, and he gets that sort of electric shock look about him. It's true.
It's very true.
And it makes me think about my mom when I was growing up, who used to do the same thing, and by the time I got to be about 10, she had a little bald spot on the top of her head. And then we children convinced her of a good mantra. Maybe your mom could tell you the same thing. And that is count to 10, say Hail Mary, and then you can say, "Why mothers get gray?" And that might help you from doing that. Okay. Without further ado, I give you Doug, who will tell us about new approaches to understanding human variation in genomes.
Well, Celeste, that was easily the most generous introduction I've ever had. Thank you so much.
Now he's going to pull his hair out.
Right. I'll have a couple of things to say. First, the tenure thing, I hope it's not jinxed. There's a long way to go yet, but it's good to know that signs are positive. Kate, who's a research scientist in my lab, recently made this same point about the pulling out of the hair. And I did go for a run just before this talk and take a shower, so the state of my hair has been randomized, and you'll never know how stressful today was. Anyway, your introduction was a really great setup for what I hope to accomplish tonight. It's true that I trained as a chemist and have come to genetics and genomics later in my career. But I thought it was, so even despite that, I thought it was, and we'll talk a lot about proteins tonight, but I thought it was only fair, since you all have been generous with your time to come listen to my talk, to start with a DNA sequence as the sort of first thing to see. And so this is a sequence of DNA, right? A's, T's, G's, and C's strung together. And the eagle-eyed among you may have noticed that this sequence happens to start with 3 letters ATG and end in 3 other letters TGA. And it turns out that those letters cause inside cells this sequence to be translated into a protein. And this is no random protein. So this is the sequence of amino acids that is encoded by this DNA sequence, as you pointed out. And this particular protein is no random protein. It turns out to be a protein called apolipoprotein E, which does in fact relate to Alzheimer's disease and is a story that we'll get into later in the talk. But I will reiterate a couple of the things that Celeste said, which is that these protein coding regions that are found inside genomes are really special because they encode these sort of workhorse molecules, proteins, that go out and sort of help all the chemical reactions in the cell that happen and make up the structure of our organs and our bodies. And so these regions are special, and we find them in every genome that we look in, from bacteria to human beings. And what's also true about these very special, so there are about 20,000 of them in the human genome, by the way. What's also true about these regions of the genome is that sometimes they change, right? And so I'm illustrating one particular change in the DNA sequence here to a T. And some of those changes, which I will from here on attempt to refer to only as variants, but I may slip up and call them mutants. So forgive me if I do that. But some variants, like this one, are special in the sense that they not only change the DNA sequence that encodes the protein, but they change the sequence of the protein itself, here to the letter W, or a tryptophan. And it turns out that these protein coding changes are, as Celeste pointed out, important for defining the differences between all of us in this room, and can also have profound consequences for disease. And that's what I want to spend our time together talking about. So it's unclear how far we'll sort of get through the talk, because I hope you all feel free to raise your hands or even interrupt me and ask questions. But the plan for tonight is to go through an example of how changes in DNA sequence can bias our risk for disease, Alzheimer's disease in particular. And I'm going to attempt to tell you how a geneticist would approach the problem of identifying that link between a genetic change and a disease risk. And then I'm going to tell you why that method, that sort of standard genetics method, is not going to work in the context of all the massive pile of variants like this one that we find when we sequence a human genome. So I'll try to give you a sense of the scale of the problem, and that will set me up to tell you about the solution that my lab and other labs are pursuing based on this technology that we developed. So that's the plan, and if we have time, I'll get into one or possibly two projects that are going on in my lab right now that deal with these matters, but we may or may not get there, so we'll see. Okay. So Alzheimer's disease is the example that I chose, and I chose this example for a number of reasons. The first is that it's something we can all relate to, right? Most of us know that Alzheimer's disease is a neurodegenerative disease that impacts primarily the old, and I'm showing you just a degenerated Alzheimer's hemisphere of a human brain compared to a sort of normal-looking one. And additionally, those of you who were at the talk last week will have heard my colleague Matt Kaberlein talk about aging. And it turns out that Alzheimer's disease is linked to aging. In fact, aging is the biggest risk factor for Alzheimer's disease. But it turns out that genetics is another really important risk factor for Alzheimer's disease. And we'll talk about this as an example of how variation in the genome can influence disease risk. Now, if we want to get a sense of the fact that Alzheimer's disease has a genetic component, we can think about what happens when a twin, an identical twin, or when their twin develops Alzheimer's disease. And in that case, there's about a 50% chance that the unaffected twin will go on to develop Alzheimer's disease. Now, you could compare this to non-twin siblings, where the chance is only about 25%. So what that tells us is that there's a significant genetic component for Alzheimer's disease. The challenge then was picking apart exactly where that risk is in the genome, right? Like which genes in the genome confer that risk. How do we know that? How do we find them? How do we prove it? Well, the other reason that I chose Alzheimer's disease as an example, and I guess I'm not going to tell you one of the real reasons, which is that I also studied Alzheimer's disease, which Celeste told you about, so that was my selfish reason. But another reason why this is a great example to talk about is that it turns out that a lot of the genetic risk for Alzheimer's disease adheres to a gene called apolipoprotein E, or ApoE, which is the same one that I showed you on the first slide. And the story of how apolipoprotein E variants in apolipoprotein E relate to Alzheimer's disease risk is actually a story that started in Seattle. So researchers both at the Seattle VA and also right here at the UW in the medical genetics and I think pathology and biostatistics groups, but I'm not sure, were the ones who published the work I'm just about to tell you about in the late 80s and early 90s. And so the way that they figured out that there was a variant of ApoE that conferred a risk for Alzheimer's disease was they went out and found 51 individuals who were affected with Alzheimer's disease. And in those individuals, sort of measured the frequency of a particular variant of ApoE, so a particular set of changes in the ApoE gene, which I'll tell you about exactly what they are on the next slide. But what they found in this group of 51 individuals was that that particular variant was present in about 60% of the individuals who had Alzheimer's disease. Okay, you might say, What does that mean? Is 60% a lot? Is that a lot of people or a small number of people? Well, they needed a control. They needed a group to compare these Alzheimer's disease-affected individuals to. So they did what I think was logical, and they went out and found 226 unaffected individuals, so healthy individuals, or at least non-Alzheimer's disease-afflicted individuals. And they measured the frequency of the very same variant in those 226 people. And what they found was something very different. They found that the variant was present in only 40% of those unaffected individuals. And they concluded, probably the same thing that you're all concluding in your brains now, which is that this variant must be associated with Alzheimer's disease because a much larger fraction of the people with the disease have the variant as compared to the people who aren't affected with the disease. So this type of study is called an association study because it establishes the association between the variant and the disease risk. And this is that sort of workhorse method of genetics that I was telling you about that's been used many times over the years to establish linkages between a particular variant in the genome and the risk of a disease. So that's sort of the first important point to appreciate. Now, over the next sort of 20 or 25 years, we've come to know a lot more about variants in ApoE and how they affect Alzheimer's disease risk because many researchers have examined this risk in very large populations of people, bless you, and we now know that there are three variants of the ApoE, three main variants of the ApoE gene, the E2 variant, the E3 variant, and the E4 variant. And just like I showed you on the previous slide, the ApoE4 variant, which is the one that our Seattle researchers were studying, though they didn't call it that at the time, is present in a fairly small percentage of the normal population, but a fairly large percentage, about 37%, of the Alzheimer's disease afflicted population. And so what this finding allowed people to do was basically determine how having or not having the ApoE4 variant, or being a carrier, in sort of technical terms, affects your risk of developing Alzheimer's disease. So it's true that among ApoE4 non-carriers, there's approximately 20% of them will go on to get Alzheimer's disease at some point in their lives. And the age of onset's pretty late, it's 85. Compare that to individuals carrying 2 copies of this disease-associated allele, and you find that 90% of them will get Alzheimer's disease at some point in their life. And the age of onset is much, much earlier, 68. So that sort of tells the story of how variants in ApoE are related to disease. At least it's part of the story. So what I'm going to tell you about now is the rest of the story. So another way to look at these alleles, variants, it's a technical term that I wasn't going to use, but that these variants, another way to look at these variants is to think about how many times they occur in a group of individuals, 150,000 individuals, who've had their genome sequenced. And these people's sequences, de-identified of course, are available in a public database. And what these circles show is the number of times you see the different alleles, the different variants, in this database. So here's one that's present 100,000 times, 27,000 times, and 10,000 times. So these are what we would call common variants. They occur frequently in the population. What's true, though, among those 150,000 individuals in the database is that there are a lot more variants that have been observed, but many fewer times. So each number here just tells you sort of the number of times a particular variant has occurred. And what you'll notice is that there are many, many variants that are observed in this population of 150,000 people only once or twice. These variants are variants that we call rare because they're rare in the population, right? We don't find them very frequently. And because we know, right, that the common variant, that one of the common variants of ApoE can confer an increased risk for Alzheimer's disease, it's reasonable to ask, what do these other variants do, right? All these other rare variants, do they confer an Alzheimer's disease risk? Should a person who carries one of these variants be worried or not? And what I hope you can appreciate is that the trick that we played before, the association trick, we can't play that trick for any of these rare variants, right? The conceit of that method was that we would find a bunch of people with Alzheimer's disease and a bunch of people without, and we would compare the frequency of the different variants in those people. But these variants are so rare, as of now we've only seen the ones that have a 1 here, just in just one living human, that we can't build up enough examples to determine whether there's an association. So we're stuck. And the problem is, in fact, even worse than that, because there are an additional 2,500 variants of the ApoE gene that could occur. There are changes in the DNA sequence that could happen, but we haven't yet seen them. So any one of these variants could be out there in the population. And we'll talk about these unseen variants in a minute. But I want to generalize now from ApoE to all the disease-associated genes in the human genome. There are about 4,000 of them. And what I'll show you is the same pattern that we saw with ApoE adheres to all the disease-related genes in the genome. So in all those 4,000 disease-related genes, we observe, in the 150,000 people who have been sequenced, 900,000 variants. That's a lot, right? What's true is that only 50,000 of those variants have been what we call interpreted. That is, we've sort of said, we know that this variant is either associated with disease or not, or said another way, pathogenic or benign. So that leaves a gap, right, of 850,000 variants that we know are out there in human beings that are in disease-related genes, but whose effect we're uncertain of, which is, was there a question? No? Okay. That's a big gap already, right? However, there are an additional approximately 11.5 million possible variants in these 4,000 disease-associated genes. So this is the pile, or you could think of it as an ocean, of variants that you find when you go out and sequence human genomes, right? Millions and millions and millions of them. Well, a million that we've seen so far, and millions that we could see in the future, yes?
And this is just in the 4,000 genes that are related to diseases that we know are related to diseases, right?
Correct. I've made an approximation here to get these numbers, and I'm happy to talk with you about it, but they should be pretty close. Yeah, so there's the other 16,000 genes, and the question, what do those genes do, is a good question that I'm not going to address in this talk, and that's not my research. I don't have the answer to that question, but it's a good question. We can talk about it offline. So what you're probably wondering now is, okay, you've shown me this big light blue box of possible variants. Should we care about them? Do they matter? I mean, after all, these are the million that we've observed. Shouldn't we focus on those? Well, what I want to take you through now is a back-of-the-envelope calculation, okay? And that back-of-the-envelope calculation starts off with a fact, and that fact is that every time a new human being is born, they carry about 60 what we call de novo variants. That means variants that were created as that human being was created. So variants that are present in the new human being, but not mom or not dad. So every time, each of us, everyone who's ever been born, has, on average, 60 new variants, 60 de novo variants. And because we know that there are about 7 billion people alive on the planet today, each of whom carry 60 variants, new variants, then we know that there must be at least, or about, 50 instances of every possible variant in the human genome out there walking around today, except for the ones that are incompatible with life. Yes?
Wouldn't it have to also be the case that any one single individual must have a very large number of variants just within their cells?
Oh, yes.
No.
Oh, yes. So there are about 4 million or so variants that are shared, that are what are called common, that I was talking about before, that are shared amongst many of us alive today. There's a burden, and I may get the numbers wrong, but I'll be in the right ballpark, of about 1,000 or so additional what we would call rare variants, right? Ones that are at very low frequency. And then layered on top of those, or included in that group, would be these 60 de novo variants. But you're absolutely right. By no means are there only 60 differences. These are just the 60 differences that are a consequence of the human being generation process, a direct consequence.
I'm raising a slightly separate issue. That just as a matter of just cell copying among how many cells that bear a DNA in a nucleus does the human body have? Is it order of magnitude a trillion?
More, tens of trillions, I think.
Okay, then that is much more than the 7
Oh, yes.
And so surely there are vast numbers of variants just from cell to cell within one person.
Vast. And this gets to a question that we may or may not have time to address, but the question of variation that's what's called in the germline, that is present in every cell in a person's body, or in somatic tissue, right? So there might be only one or a few cells that have that particular variant that was generated when the cell divided, right? And it turns out that those somatic variants are the ones that can be involved in cancer. Right? So when we look in cancers, we also see a large number of variants. So your point is well taken, and we'll see if we get there, but tonight. But the point that I guess your observation underscores is that every variant that could occur in the human genome is out there in the population, and I suppose if we think about it, in every one of us in one cell or the other, and each one of those variants will be generated again when a new human generation is born. So the answer to the question, should we worry about all those possible variants that we haven't seen yet, is yes, we should, because they're out there, and if we go sequence human genomes, say the genomes of all the people on the planet, we'll find them. Okay. So at this point in the talk, I want to spend one slide, and I'm only going to spend one slide, talking about whether knowing the effect of all these variants is meaningful. And I picked the ApoE example. The final reason I picked it is that I hope it's really relatable, right? Like, I happen to know that I'm a carrier of one copy of the ApoE4 allele. I find that to be useful information to know. I think people may react differently, whether they think it's a good idea, like they would want to know or they wouldn't want to know. I can tell you for certain that insurance companies, many of them would like to know, although legally they can't take action on that information at the moment. So knowing the effect of variants in ApoE, that's a really fraught question. And thinking about it for a little while reveals all the complexity about the challenges of learning your own genetic information when there's a meaningful difference on the line, and of what might happen in society as this information sort of unfolds. But, so we can have discussion about that, and should. I do want to give you just a couple of additional examples to sort of whet your appetite. There are a number of cancer-related proteins, like TP53, BRCA1, which is also discovered, related to breast cancer, and also discovered here at the University of Washington, and PTEN, which we may have time to talk about later. So variation in these genes can predispose a person to certain types of cancer, or if variation in these genes occurs in somatic tissue and creates a tumor, it can change the aggressiveness of the tumor or how the tumor might be best treated. There's a group of genes referred to by the buzzword pharmacogenes, because these genes control the body's metabolism of drugs. And so variation in these genes can change how a person responds to a drug, or even whether they have a toxic side effect or not. So knowing the effect of these variants can be really meaningful if you're, say, someone who would have a really bad toxic side effect to a drug because of a variant that you carry. And a final example that maybe is one that we're most familiar with, is that there's lots of variation that's linked to inherited disease, like, for example, cystic fibrosis or sickle cell anemia. And so you may very much want to know whether you're a carrier of a pathogenic variant in one of these genes, because you could use that information to decide or know, for example, like what risk your offspring might be at for such a disease. So these are just a few examples, but the sort of take-home point to appreciate is that of the 4,000 disease-related genes in the human genome, knowing the effect of variants that a person carries in many of them, certainly not all, not even most at this point, but many of them, is really useful information, both in the clinic and perhaps personally. Okay, so that's sort of the first part of the talk, and I hope I've succeeded in sort of setting out the problem for you. There are a huge number of variants in the human genome that are out there. We can't interpret them. We can't figure out what they do by association, right? By this sort of classical genetic approach. So we're left with the question, how do we interpret these variants? What do we do with them, right? Or said another way, how do we establish the relationship between the variant and the phenotype which is sort of just what happens, what we observe in someone carrying the variant. So that could be a disease risk or a change in hair color or eye color or anything else we can observe. So what I'm going to argue today is that going into the lab and testing the effect of the variant in what we call a functional assay is one way to solve this variant interpretation problem. And when I say variant interpretation problem, I just mean big pile of variants, traditional methods won't work. Okay, so rather than try to explain what a functional assay is in words, I thought it would be useful to just show you an example of what I mean. And so one example of a functional assay is to measure the growth of a cell, of a culture of cells, carrying a variant. Because for many genes in the genome, cell growth sort of depends on those genes. And if you mess up the function of one of those genes, then the cells won't grow anymore. And this will become clear in a second. But here's an example culture of a particular wild-type sequence of a gene growing, or whether the cells are growing, harboring this wild-type sequence. And you'll have to just trust me, but this is a normal-looking growth curve, okay? So we're recording how many cells there are over time, and the cells divide and multiply and grow, right? Now, if we replace the wild-type copy, the normal copy of this gene, with a variant, we can then measure that variant's growth. Oh, and the red line has, I guess, ended up under the black line. But what I hope you can appreciate is that this variant, it grows just like wild-type, right? So this is called a growth curve, and these two growth curves, they look the same. Now, we could test a second variant in the lab, this blue variant, and we can see this one looks very, very different, right? The growth curve here is flat, meaning these cells, they don't grow at all. And so what we know, what we've done, this operation, is going from the variant, right, these guys, to the functional effect in the lab. Grows just fine, doesn't grow at all. Yes?
Are there ever curves that grow faster? Or is it always that they do the blue thing if they're bad?
Indeed there are, and that is a really deep what happens then is a really deep question. So some variants, right, what we're talking about here are proteins. You can tweak the function of a protein and make it, say, a little bit better at catalyzing a reaction. And maybe that makes the cell grow a little bit faster in the lab, right? And the deep question is, do you think that just because the cell grows a little bit faster in the lab, is that a good thing or a bad thing for the human being carrying the variant? And that gets to the challenge of the next step we want to do, which is go from the measured functional effect in the lab to what happens to the human being carrying any of these variants, right? And in the case the simple case that I've laid out for you here, maybe it's pretty straightforward, right? We would say, well, a person carrying variant number 1, they're probably okay, right? Because their variant looks like wild type. But a person carrying variant number 2, if this were a variant in, say, a cancer predisposing gene, then we might be pretty worried about this person because their variant looks very different from wild type. Now, if we had a variant that grew faster than wild type, I would personally be worried as well, but we'd have to do a lot of further work to establish just how worried we would be. In any case, hopefully this example illustrates how we can go from a variant to an effect that we measure in the lab to a conclusion about what a human being who carried that variant, what we might expect to observe in that human being. Okay, oops, I've gone back. Let me go forward. Now, I showed you 3 test tubes that we were gonna make and measure in the lab, and those would literally be physical tubes, right? That we would take to a machine, we'd measure how many cells there are, we'd put them back in an incubator, and we'd repeat that process over the 36-hour growth curve or 48-hour growth curve. So you could imagine if you were very industrious doing that for maybe 100 variants, 100 test tubes, 1,000 variants, 1,000 test tubes, but you certainly couldn't do 10,000 variants, right? That's too many. Well, it turns out that 10,000 variants is the number of variants, the number of possible variants in one human gene. There are about 10,000 possible variants. So even getting to sort of understanding all the variation in just one gene, it's impossible to do it one at a time, right? There are just too many things to measure, too many test tubes. And so the solution that we've devised to this problem, and when I say we, I really mean we and Stan, who's there, that we devised when I was a postdoc in his lab, is this technology we call deep mutational scanning. And I'm going to attempt to tell you a little bit of the technical details of how this technology works. But before I do that, I want you to appreciate what the point is. The point is, rather than measuring each one of those variants separately in a separate test tube, we pool them all together. And then by pooling them all together, we're able to make measurements on, say, all 10,000 possible variants of a gene simultaneously, right? So we read out the effects of all the possible variants at once. And that's the trick. That's the increase in scale, going from one at a time to parallel measurement, right? Measuring many things at once. So what I'm going to do now is attempt to tell you exactly how that works, and we'll see how it goes. So feel free to stop me if it becomes, if I mess up. OK, so the first thing we do is we take the DNA sequence encoding the gene that we're interested in, and we make a library of variants. Now, I'm only showing you a library of 3 variants here. Our familiar red one, the wild-type sequence, the black one, and the familiar blue one from a few slides ago. The real libraries we make would have, as I said, all 10,000 possible variants in a protein. Next, what we do is we sort of insert those variants into cells, right? Just like we did in the previous example. But again, instead of having each culture of cells sort of be separate, we're mixing them all together, literally in one physical flask. So now we have our wild-type cells growing together with our blue cells and our red cells. Now, if you were paying attention a few slides ago, and if I did my job correctly, which I may or may not have done, you know what should happen next, right? If we let these cells grow, the black ones and the red ones are going to grow just fine, and the blue ones aren't, right? So even though they're all pooled together, we still have the same phenomenon going on. The black ones multiply, the red ones multiply, but the blue ones don't multiply because the blue copy or the blue variant is defective and it can't support cell growth. OK, so we've got these cells mixed together. What do we do next? Well, we turn to the most powerful method in modern genomics, I would argue, although some might disagree with me, and that's DNA sequencing. So we now have high-throughput DNA sequencers that are capable of literally sequencing every cell in this population and determining whether it's a black cell, a blue cell, or a red cell, and every cell in this population, and again, determining black, red, or blue. And so what we've done is count the number of times each of our variants occurs at the beginning and at the end of the growth, right? And so we can just say, aha, well, I see that these black cells sort of grew OK. I counted 4 of them in the beginning and 6 of them in the end. And the red cells, I counted 3 in the beginning and 6 in the end. But the blue cells, I counted 3 in the beginning, but only 1 at the end, right? And that process of counting allows us to establish how each variant functions relative to wild type. It lets us do our test tube at a time experiment all mixed together. So we can score each variant. So red gets a score of 1 because it's just like wild type, whereas blue gets a score of 0.1, right? Because it's much worse than wild type. It grew much more slowly. So that's the trick. The trick is using high-throughput DNA sequencing to count, and then from the counting, figuring out what happens. Yes?
Do you ever worry about the various lines competing with each other?
That's another excellent question. So the question was, do you ever worry about competition for resources, like, for example, sugar, say, in the media, the growth media, between the different cells? And the answer is yes. You have to make sure to do these experiments in such a way that there's not competition between the cells. And you can do that by making sure that there's plenty of resources around and you don't grow the cells for too long. But if you don't do that, then you see really weird data that doesn't make a lot of sense, and so you've got to be careful. OK, so where did we get? Well, what we get, or what do we get? At the end, we get something that we call a sequence function map. And it's this data that we argue is going to be so useful in solving the variant interpretation problem. So I'm going to teach you how to read a sequence function map. And it's read as follows. So each column in this map is a different position in the protein. So this is the first position, the second position, the third position, and so on. And I'm only showing you a little chunk of a sequence function map that covers about 20 positions. A normal protein would have 350 positions, so it would be way too big to show you anything. So I'm just showing you a little chunk here. Now, each row is a different possible variant. Okay, so this row is a tyrosine variant. a tryptophan variant, a phenylalanine variant. Those are different amino acid substitutions, right? Different variants. And so when you look down a column, that's showing you every possible mutation, the effect of every possible mutation that could occur at that first position in the protein. And collectively, across all the positions and all the possible substitutions, what a sequence function map does is reveal the effects of every possible mutation, or every possible variant, sorry, there's another word I wasn't going to say, every possible variant in the protein, right? And we read these out as ones that are red being wild type-like. So again, just like our example, those are ones that grew just fine. And variants that are blue, those are what we call loss of function or inactive variants. Just like in our example, the blue ones don't grow at all, right? So the red variants are the ones that we would say, okay, we're not so worried about a person carrying this particular variant. The blue ones are the ones we would say, yep, we might worry. But the reason why we think this data is so useful is that it solves this problem of the massive number of possible variants, because we've made a measurement in the lab of what each one of those variants does, right? And I told you how we can make an inference based on what those variants do in the lab we can make an inference about what they might do in a human being. So once we've collected one of these maps, then for any future variant that we observe in a human genome, or any of the variants that we've already observed, we can just go and look up what it does, right? So we've solved the variant interpretation problem in that way. At least that's the idea. So what my lab has been working on over the last 4.5 years is really fleshing that out, right? And when we started with that first map that I showed you was pretty much the first one we made, and it was just a little chunk of a protein. So we had to learn how to make a map for the whole 350 amino acid long protein. We had to learn how to do these experiments in cultured human cells because we wanted to be able to make very accurate inferences about what would happen to humans carrying these variants. And so I'm going to spend the next hour, no, I'm just kidding, telling you about 2 projects in the lab. I'm not going to spend any more time, or much more time. I'll take a few minutes to summarize. So you can ask me about these projects if you like, but in the first one, what we did was use deep mutational scanning to figure out all the drug resistance variants associated with a cancer-related protein called SRC. There's a drug that inhibits it, and we found all the resistant variants that could ever happen in a human being in this protein. We're trying to figure out how to use that data in the clinic. And then in a second project, what we're trying to do is scale this approach genome-wide. So for all of those 4,000 disease-associated proteins. And to do that, we had to get really smart about the kinds of experiments we were doing in the lab. Because before, we'd been sort of, I guess the analogy would be we'd been making these bespoke tailored suits, right? Like we'd find the protein and we'd craft an experiment just for that protein. And that worked great when we only wanted to look at 1 or 2 proteins. But what this project's all about is like going to China, buying the factory, and making sure we can make experiments for all the proteins in the genome. I mixed my metaphor there. We are not actually doing anything in China. I should be careful with my metaphors. I'll get in trouble. But maybe you get the idea. Okay, so I'm going to zoom past all these slides that I included and didn't need, this is another thing that as an assistant professor you're getting accused of, which is trying to show too much data, which is certainly a thing that I was going to do, but thankfully I've saved you from that fate. And I'll just end here on the vision, right? So our vision is to construct what we're calling an atlas of functional or experimental data for the human genome, where we would like to know the effect, at least in the lab, of every possible variant in every disease-associated gene in the human genome. And that sounds like a really grand goal. It is a really grand goal, and it's not just us doing this. So we're pulling together a coalition of researchers, an international coalition of researchers, to try to tackle what we view as the sort of next critical challenge in genomics. Because now we can sequence genomes very cheaply, right? Well under $1,000 to sequence a whole genome. And the next task is figuring out what can we do with the information in those genomes. And that task, we argue, and I've tried to convince you tonight, reduces to understanding what the variants in those genomes do. And we think our method is a way forward. So I'll just take the last couple of minutes and thank the people who are involved in the work. I'm very fortunate to have a really spectacular group of people to work with. I have no idea how I got that lucky. it's truly one of the best things about my job is working with this group of people. So basically all of them on this slide contributed to the ideas that I told you about. The SARC project, which we zoomed through, is the work of Ethan. He's a graduate student in the lab, and he's worked with Jason and Kate, as well as Akena, an undergrad in the lab, to get that project done. It's very exciting. We're writing the paper right now and hope to submit it in the next few weeks. So we're really excited to see what sort of the community thinks about it. Kenny Matrejek is the postdoc in the lab who, along with Leah Storita, a collaborator in Jay Shanduri's lab, figured out how to build the factory in China, for lack of a better metaphor, and allow us to assay, we hope, most of the genes in the human genome in the way that I told you about. He also worked a lot with Jason and a few others in the lab. I'm very fortunate to have funding from the National Institute of General Medical Sciences, so that's a part of the NIH. And if you like sort of biology research, you should know that NIH budget is under threat, and you should call your Congress people. Same with the Pharmacogenomics Research Network. And then I also am supported by the Alzheimer's Association for some work that I didn't talk about. So with that, I suppose we have about 10 minutes or so. I can answer questions in a sort of public forum, and then we can break up and talk.